After comprehensively discussing on the thyroid gland, we are moving our focus towards the adrenal glands and the gonads, where we'll be discussing on the adrenal and the reproductive endocrinology. We are not going to discuss on the core anatomical principles of the adrenal glands and the gonads because they have their own anatomical and surgical relevance, and they will be discussed in the relevant surgical specialties. And discussing about the core anatomical principles and the relationships of each and everything is not important to understand your basic endocrinology and the disease associated with endocrinology. And I don't want to dilute by telling all those things. So without wasting much time, let us move on to the parts of the adrenal gland. Adrenal gland is basically divided into two parts. One is the glandular part and second one is the non-glandular part, which is nothing but the neural part. This is very common to many other endocrine glands, where your thyroid also has a glandular part in the form of thyroid epithelial cells that secrete thyroid hormones, and non-glandular part in the form of parafollicular cells, otherwise called as C cells, which secrete calcitonin. At the same time, your pituitary gland also has a glandular part in the form of adenohypophyse and neural part in the form of neurohypophyse, and pancreas may be having an exocrine pancreas and an endocrine pancreas. So this is not something very new to the adrenal gland, but still we have to understand this. This glandular part of the adrenal gland is what we refer to as the adrenal cortex, which makes up 75% of the gland. And the neural part of the adrenal gland is what we refer to as the adrenal medulla, which makes up to the remaining 25% of the gland. You know where this glandular part comes from, or otherwise the cortex comes from? It comes from the urogenital ridge, which itself is a part of intermediate mesoderm. Clear? And you know where your neural part comes from? Neural part has a different origin. It comes from neural crest cells. These neural crest cells differentiate into some specialized group of cells called as chromaffin cells. These chromaffin cells have unique function because they are endocrine in nature. They secrete catecholamines into the blood. I'll tell you the importance. I'll substantiate in some time. But they are made of chromaffin cell clusters which have their base in the neural crest cells that come and reside in the adrenal gland to form the adrenal medulla in the first place. The cortex you can divide into three more areas. One is the zona glomerulosa and the zona fasciculata, and third one is the zona reticularis. The fasciculata forms the bulk of the adrenal cortex, contributing to 50% of the cortex, and glomerulosa contributes to 15% of the cortex, and reticularis contributes to the remaining 10% of the adrenal cortex. The total is the 75%. That's why I divide into 50, 15 and 10. So histologically, you can differentiate by just looking under the microscope. The glomerulosa will be a little different in the form of oval clusters of cells. You can see they form only a little portion on the gland, on the adrenal cortex, where they form this oval cell clusters. Fasciculata by far is the bulk of the adrenal cortex. So where you can see the cells are arranged in the form of straight cord-like structures, and when you zoom it in high power and if you see, you can note the fact that the fasciculata cells are arranged in straight cords. And you can also see in the adrenal cortex, there are a lot of these rounded white color structures in high power. So these are nothing but lipid droplets in the cytoplasm. So because adrenal hormones are, cortical hormones are steroid hormones, they need cholesterol. So this is the storage of lipids that are seen as this white roundish structures in the cytoplasm. So this is the fasciculata which contributes to the bulk of the gland, 50%. And the remaining is reticularis, which contributes to 10% of the gland, where reticularis cells are arranged in a net-like pattern. Fine. So glomerulosa, oval cell clusters, fasciculata, straight cords, and reticularis in the form of net-like pattern. What is the principal hormone they are going to secrete? The principal hormone that glomerulosa secretes is the principal mineralocorticoid, that is called aldosterone. Even though there are other mineralocorticoids that are secreted like corticosterone and deoxycorticosterone, but aldosterone is considered as the principal mineralocorticoid and the strongest mineralocorticoid by default. And the fasciculata secretes glucocorticoids, and the principal glucocorticoid that the fasciculata secretes is the cortisol. And the reticularis is going to secrete your adrenal androgens, sex hormones, and the most important adrenal androgen and the highest output is in the form of DHEA, This is called dehydroepiandrosterone sulfate. At the same time, they secrete a little bit of androstenedione also, which I'll be writing as A4, like what I have written here. So that's A4 is androstenedione. But the principal output is always DHEAS. So the principal output from glomerulosa is aldosterone, principal output from fasciculata is cortisol, principal output from your reticularis is DHEAS. So who is controlling this? You have to understand the fact this aldosterone is completely under the control of angiotensin II and potassium, which means they are completely under the control of renin, angiotensin, aldosterone system. Because it's a common mistake from a student's perspective, especially as a beginner, they might be mistaking that this aldosterone is in the control of ACTH. No, the principal controller of aldosterone is angiotensin II, potassium. In other sense, it is renin, angiotensin, aldosterone system, RAS. And the principal controller of this cortisol and DHEAS is your ACTH, which means your fasciculata and reticularis is controlled predominantly by ACTH, whereas your glomerulosa is predominantly controlled by your renin-angiotensin system. And to be honest, the feedback to the pituitary is also in the form of cortisol. Cortisol is the principal agent that gives feedback to the pituitary regarding the adrenal status, and your ACTH levels will be dependent mostly on the cortisol levels in the blood, not the aldosterone levels. So this is again a key point to understand. The adrenal medulla, we know they are formed of chromaffin cells which carry their own importance. If you ask me whether chromaffin cells are seen only in the adrenal medulla, in adults, to an extent your answer is yes, but in the embryological, in the fetal life or in the early childhood, the answer is no. Because in the embryological life and the fetal life, and in fact in the early childhood up to 1 to 3 years of age, there are other areas of chromaffin cell clusters seen in the abdomen. Because chromaffin cells, the natural habitat is towards the abdomen only. So there will be a lot of chromaffin cell clusters seen elsewhere in the, distributed elsewhere in the abdomen also. One of the largest chromaffin cell clusters, in fact, in the fetal and the embryonic life is not adrenal medulla. It is in the form of organ of Zucker-Kandel, which will be seen in the para-aortic region, which I'll be discussing subsequently in the pheochromocytoma chapter as well. But there should be some basic idea about this chromaffin cells, and you have to clearly understand the fact that these chromaffin cells are not unique to the adrenal medulla. In the childhood, all the other areas apart from the adrenal medulla will regress and involute. So that makes in an adult, adrenal medulla being the prominent and the principal source of this chromaffin cell clusters. That's it. Which means the tumors of this chromaffin cells, which means the tumors of the adrenal medulla, we call it as pheochromocytoma. Yes, that's right. But what I want to tell is the fact that these tumors of the chromaffin cells need not originate only from the adrenal medulla, right? Yes, most commonly they originate from the adrenal medulla because in adult, the principal source of chromaffin cells is the adrenal medulla. So pheochromocytomas most commonly originate from the adrenal medulla. But there can be extra-adrenal sources of pheochromocytoma as well, because the extra-adrenal chromaffin cell clusters that are seen in the fetal and the embryonic life and probably in the early childhood, usually they go for involution, but they can wake up in the adult life in the form of like tumors like pheochromocytoma. That is why there is an entity called extra-adrenal pheochromocytoma as well. So what is the importance of these chromaffin cells? Chromaffin cells have some unique functions. So the first thing I want to tell is the synthesis of one unique substance called adrenaline. In what way adrenaline is unique? Many people might be laughing on this, but still, yes, it is unique function of chromaffin cells in the body. So let us see other areas, other neural areas apart from this chromaffin cell clusters, what they are going to do. Yes, they do secrete catecholamines, but what they secrete, let us see. So you know the basic product is the tyrosine, from which you will produce L-DOPA. Of course, in this process, there are a lot of enzymes involved like tyrosine hydroxylase, DOPA decarboxylase, dopamine hydroxylase, which we'll be discussing subsequently in the physiology of the adrenal medulla. But right now, let us don't complicate and see they are going to secrete dopamine, and from the dopamine they are going to secrete noradrenaline. This noradrenaline will be released into the synapse in the synaptic terminal. So this is the presynaptic terminal and this is the postsynaptic terminal. And in the synapse they will be releasing this noradrenaline, and this noradrenaline will be acting on the noradrenaline receptors. So once their work is complete, most of the noradrenaline will be taken back into the presynaptic terminal itself. So this is how they regulate the noradrenaline release. Remember the fact that these neural structures apart from these chromaffin cells cannot secrete adrenaline. Very, very important point. That comes to the importance of the chromaffin cells. These are the chromaffin cells, and the most important point is the fact that they are endocrine, which means they are going to release their secretions directly into the blood. So you have a tyrosine which forms L-DOPA, which forms dopamine, which is going to form noradrenaline, and this noradrenaline will be converted to adrenaline as well. So this conversion to adrenaline is mediated by a very special enzyme called a PNMT. That's called a phenylethanolamine N-methyl transferase. So this is a key enzyme and a unique enzyme for this chromaffin cell clusters, which means other neural structures apart from this chromaffin cells cannot secrete this adrenaline in the first place. And the second key point regarding the chromaffin cells being endocrine in function, they are going to secrete this noradrenaline and adrenaline directly into the blood, which means, as the name suggests, most of their secretions into the blood will be in the form of adrenaline, where 85% of the chromaffin cell cluster output in the form of adrenal medullary output in adults will be adrenaline, and remaining 15% of their output will be in the form of noradrenaline. There are minor amounts of dopamine, trace amounts of dopamine are also secreted, but that is not physiologically relevant in the first place. So the most important secretion will be the adrenaline, and remember, whatever adrenaline you see in the blood, the source is only chromaffin cells. But in adults, you can equate these chromaffin cells to the adrenal medulla, because other areas of chromaffin cell clusters in the abdomen in adults would have been already involuted and lost their function. Clear? So what is the importance of understanding this in a pheochromocytoma? Because it's very important for understanding your diagnostic value, because this adrenaline and noradrenaline will be further processed by various group of enzymes, and there are two key enzymes that process this adrenaline noradrenaline, that is called a monoamine oxidase, peripheral MAO, and a peripheral COMT, that's called catechol O-methyl transferase, which is going to convert this adrenaline into a metanephrine and this noradrenaline to normetanephrine. So this metanephrine and normetanephrine will be further acted upon by the same enzymes, that is monoamine oxidase and catechol O-methyl transferase, where they will be converted to a final product called vandyl mandelic acid, which will be excreted in the urine. So that is the reason your old biochemistry textbooks will give this vandyl mandelic acid, the 24-hour urine collection, and finding out the VMA in the 24-hour urine is diagnostic of a chromaffin cell tumor. These chromaffin cell tumors are what we refer to as a pheochromocytoma. Most common location in adults is adrenal medulla, but there can be extra-adrenal pheochromocytomas as well. We discussed that already. But is vandyl mandelic acid specific? I don't think so, because they are a common product of metabolism of both adrenaline as well as noradrenaline. In the sense, this noradrenaline is also coming from a lot of other neural structures as well, so which means they cannot be specific, which means the most specific compound to be used for diagnosis, to be utilized for diagnosis in the diagnosis of pheochromocytoma is metanephrine, because we know the fact that adrenaline means it should come from your chromaffin cells only. Whatever adrenaline you see in the blood is coming from chromaffin cell clusters. There is no doubt about that, which means whatever metanephrine you see in the blood should also come from the chromaffin cells only. Whenever the metanephrines are elevated, that is the diagnostic standard currently for diagnosing a pheochromocytoma. It's not the normetanephrine, it's not the VMA that's going to be specific or sensitive for the diagnosis of pheochromocytoma. It's always your normetanephrine that is metanephrine that is very, very specific and very, very sensitive for the diagnosis of pheochromocytoma in the adults. Clear? So very, very important point. There are two important things that you have to understand. One, the principal source of adrenaline in the blood in any patient or any human being is chromaffin cell clusters, and in adults, almost all the chromaffin cell clusters lies in the adrenal medulla, which means the principal source of adrenaline in the blood in adults is adrenal medulla. And similarly, metanephrine is the gold standard currently for the diagnosis of pheochromocytoma for the same reason. And this chromaffin cell clusters, being endocrine in nature, they're going to secrete their catecholamines directly into the blood rather than other neural structures which just secrete into the synaptic terminals. Let us move on to the blood supply of the adrenal glands. The blood supply arrangement is more important than the actual blood supply, which I'll be discussing subsequently. But before that, let us understand some of the basic concepts like what are the basic blood supply of the adrenal gland. Adrenal gland is principally supplied by three vessels. The first one is the superior suprarenal vessel. This superior suprarenal vessels originate from the inferior phrenic artery, and you have a medial suprarenal vessel which originates directly from the aorta, and you have an inferior suprarenal vessel. As highlighted in the pointer, you can see it originates from the renal artery. So there are three blood supplies which are arranged in a unique fashion called a radial pattern of arrangement, which we'll be discussing subsequently. And the venous drainage, as such, it's common for the gonads and the adrenals because the right adrenals and the right gonads will drain into the IVC, and the left adrenals and the left gonads will be draining into the left renal vein in the form of adrenal suprarenal veins and the gonadal veins. In males, we call testicular vein, and females, we call it as ovarian veins. That's it. So right side to IVC, left side to left renal vein. And nerve supply, as such, the neural supply, the sympathetics come from the celiac ganglion in the form of splanchnic nerves, and the parasympathetic supply will be in the form of vagus. We know that, these are very simple things. And as far as the lymph node drainage is concerned, the right suprarenal gland will drain into the caval nodes that near the IVC because they are closer, and the left suprarenal gland will be draining into the aortic nodes because they are closer to them. Simple. But even though I told the basic anatomical distribution of the blood supply, but the pattern of arrangement of this blood supply is clearly important as far as adrenals are concerned. Let me draw the cortex and the medulla, where I'll be highlighting the cortex in probably green and probably the medulla in blue. So let us see how the blood supply will be arranged. The blood supply usually is arranged from all the sides in the form of radial arrangement. So where the blood supply will always be from the cortex towards the medulla. Why you need this kind of blood supply? Because as I told you, in adrenal medulla, there is one very unique enzyme called the PNMT, which converts this noradrenaline into adrenaline. So this conversion of noradrenaline to adrenaline by PNMT is completely cortisol dependent, which means they need cortisol for this enzyme's action. So that is the reason why the blood supply is arranged in a radial manner, so that you know that you have glomerulosa, fasciculata and reticularis. In the fasciculata are going to produce cortisol. And if the blood supply are arranged in a radial fashion or coming from the cortex towards the medulla, whatever cortisol that is generated in the zona fasciculata will be washed off by this blood towards the medulla, where they will be stimulating this PNMT enzyme to produce adrenaline, which means the PNMT enzyme is cortisol dependent, and without the cortisol coming from the fasciculata, you cannot produce this epinephrine or adrenaline in the first place. So that is also one reason why your epinephrine or otherwise adrenaline synthesis in the body is based on cortisol. So this is one reason why cortisol is called as permissive hormone, or this is the permissive action of cortisol, where cortisol allows epinephrine to form. At the same time, cortisol allows the epinephrine to act at a cellular level. At a cellular level, we know that this catecholamines, for example, adrenaline, is going to act on the cell surface receptors, whose signals will be translated downstream in the form of signal transduction, and they will be giving some cellular effects. So that is the action of any cell surface related hormone. But you have to understand the fact that since cortisol acts in the nucleus, in the DNA domains, so this cortisol is very, very important to be present in the nucleus to stimulate certain genetic elements to produce these proteins that are responsible for this signal transduction, which means without this cortisol, you cannot transduce the signal downstream into the cell that will translate into effects of catecholamines, which means without this cortisol, your adrenaline becomes a zero inside the cell as well. First, without cortisol, your adrenaline production drastically drops. At the same time, without this cortisol, even if adrenaline or noradrenaline is present, their effects will not be transmitted into the cell, so that whenever you don't have cortisol, even in the presence of catecholamines, you will go for shock, and that is the base of adrenal crisis and adrenal insufficiency. This is what we refer to as a permissive action of cortisol in the first place. So there are two areas where this permissive action is important. One is at the cellular level, one is at the synthesis level, where cortisol is very, very important for the synthesis of adrenaline in the first place. Let us move on to the embryology, because a comprehensive understanding of embryology is really important to understand the future topic on disorders of sexual differentiation, which will be discussed in the subsequent videos anyways. So embryology wise, there are a lot of events that are happening, which is quite complicated, but you have to know there are three things that originate from a common area called urogenital ridge. This urogenital ridge generally develops around 4 to 6 weeks of embryonic life. For exam perspective, you can remember as 4 weeks where your urogenital ridge will start. And from the urogenital ridge, you produce adrenals, and there will also be formation of kidneys and as well as the formation of your gonads. The gonads at this point, even though they are indifferent gonad or a bipotential gonad, which we will be discussing subsequently, but there are three structures that are developing from this urogenital ridge. These are very, very important. And what really happens is the kidneys are formed initially lower down. You can see here the kidneys are formed initially lower down, where there will be having ascent subsequently in the fetal and embryonic life. So the kidneys come to a definitive lumbar position below the suprarenal glands. And the gonads will generally tend to descend down, especially if it's a case of a male, where on the effect of testosterone and insulin-like factor 3, they descend down and come to the definitive location in the scrotum. So there is a renal ascent and a gonadal descent. But the suprarenal glands are more or less located in the same position. They don't go up or down, usually through the journey in the embryonic and fetal life. So the formation of gonads and adrenal gland has a lot of basic understanding, and many transcription factors are involved in it, and all of them are key in understanding your disorders of sexual differentiation. So first, let us see, there is something called a urogenital ridge which develops around 4 to 6 weeks mark. From this, you are going to result in three structures. One is the adrenal gland, and second is the gonad. This is an indifferent or a bipotential gonad, and you are also going to result in the formation of a kidney. So these are three basic structures I told you that will be developing from the urogenital ridge. So now, there is one important transcription factor called NR5A1. This is a very important factor, this NR5A1, which is important for the development of the adrenal gland and the gonad as well. This NR5A1 is also known to be important for the development of the pituitary as well. The other name for this NR5A1 is also referred to as steroidogenic factor 1, otherwise called as SF1. That is the reason whenever you have a defect in NR5A1, you will be resulting in gonadal dysgenesis, you will be resulting in adrenal gland issues. At the same time, you will be having agenesis of the pituitary gland as well. So whenever a patient is coming with a disorder of sexual differentiation due to gonadal dysgenesis and they have adrenal gland and pituitary gland involvement, your problem must be, in exams at least, should be localized to the NR5A1. Similarly, there is another gene and a protein called WT1, and this is important for the gonadal development. At the same time, this is important for the kidney development also. To an extent, it's important for your adrenal gland development as well, but more important for gonads as well as kidney. So I can write gonads as well as for kidney, you need this WT1. So that is the reason whenever you have some issues with WT1, you will be resulting in gonadal dysgenesis. At the same time, you will have some kidney issues like mesangial sclerosis and nephrotic syndrome. Remember, this WT1 issues is one of the important causes of this congenital nephrotic syndrome. Clear? So this is a very important entity. So these are the basic level factors that are regulating your gonadal and adrenal development. So now let us focus more on the adrenal gland and the gonads. Your adrenal gland development further will be based on a lot of factors, but more importantly, you have PBX1, WNT5. So many of the other transcription factors will result in further maturation of the adrenal gland to a structure called as adrenal primordium, and the sustenance and the further development of the adrenal primordium is dependent on a variety of other factors like your insulin-like growth factor 2, your transforming growth factor 2, and at the same time you have POMC, which is the most important. As far as I know, this POMC becomes the most important as far as adrenal gland is concerned. This is nothing but the ACTH, pre-opiomelanocortin. It's a larger molecule which is broken down into a smaller molecule called ACTH. Remember that ACTH is the first pituitary hormone to be secreted in fetal life, which you have seen already in the pituitary basics itself. And finally, you will form the mature adrenal gland, and the sustenance of this mature adrenal gland and the most important factor that causes the sustenance of this mature adrenal gland is the POMC. Now, the POMC becomes the most important, where POMC is important for increasing the cell number as well as cell size, as well as the increase the enzyme content within the adrenal gland cells. So, POMC is the most important as far as we know. So, this adrenal gland in fetal life is different from what you see in the adult life. So, as far as the fetal life is concerned, let me tell you the adrenal gland in the fetal life first, and let me tell you what happens in childhood, especially immediately after birth in the first 2 to 3 weeks, and what will be the case in the adults, especially during puberty. Clear? So, what will be the fetal adrenal gland? The fetal adrenal gland is usually differentiated into 3 different zones. This initial zone is called as a fetal zone. This is the most active zone as far as the fetal adrenal gland is concerned. And next you have an intermediate zone. The function of intermediate zone, we have no idea. And we also have this definitive zone, which we know the function is very limited as far as we know, and the fetal zone is the most important. So let me shade the fetal zone in red and the intermediate zone in blue and probably the definitive zone in green. So as I told you before, this fetal zone is the most important in the fetal life, and we have not discussed yet the adrenal steroidogenesis, but when I am going to discuss about the adrenal steroidogenesis, you will be understanding this much better about these key enzymes. This fetal zone has more of a 17-hydroxylase and lyase activity and less of a 3-beta HSD activity. So this will definitely translate into an important entity called they are going to produce more of DHEAS, which means the principal adrenal hormone that is produced in the fetal life is DHEAS. That's very, very important point again. Many of the times asked question in the exam as well. The principal hormone that is produced from the adrenal gland in the fetal life is DHEAS, and this is the reason for why they produce excess DHEAS. And because of the low 3-beta HSD, which is very important for producing cortisol and aldosterone, they're going to produce very less cortisol. But remember, in the fetal life, especially this fetal zone will be producing more of a cortisone than a cortisol. Cortisone is much less active compared to cortisol, but they're going to produce only very less amounts of cortisone, which is actually inactive compared to that of the cortisol, in fact. So this excess DHEAS that is produced by the adrenal gland will be going to the placenta, where will be acted upon by an enzyme called placental aromatase. And this placental aromatase will convert this DHEAS to estriol. Obviously this is the androgen, and aromatase is going to convert an androgen into a estrogen. So once they convert into estriol, so they go to the maternal circulation. So please understand the fact that almost 50% of the estrogens that you see in the maternal circulation is coming from the DHEAS as a substrate. So which means the fetus is the source of the maternal estrogens. That's a very, very important point. So almost 50% of the circulating estrogens are actually coming from the fetal DHEAS. So fetus are considered as the factories of this maternal estrogens. So this brings down to one of the important clinical points where you have a deficiency of this aromatase in the fetus. For example, you don't have this aromatase and there is an aromatase deficiency. Whenever you have an aromatase deficiency, the conversion of this DHEAS to estriol will not happen, and this excess estriol will not be produced, and there will be excess DHEA. Instead, this is an androgen, and this will be circulating in the mother, leading to maternal virilization. So whenever you encounter a term called maternal virilization during pregnancy, this is a very important key point. So whenever you consider disorder of sexual differentiation, if you have a point of maternal virilization, if there is a history of maternal virilization during pregnancy, that straight away is a pointer for this placental aromatase deficiency or simply an aromatase deficiency. Clear? That's a very important clinical point. At the same time, we don't know what is the function of this intermediate zone, but we have this definitive zone which tends to produce cortisol, but they produce cortisol only during that narrow period of that 8 to 12 weeks. This 8 to 12 weeks window is really important for external genitalia formation. During this time, the fetus should not be exposed to unnecessary excess androgens. Normally itself, the adrenal glands are going to produce excess DHAs, and this excessive DHAs should not be produced during this 8 to 12 weeks mark, because otherwise, if it's a female child, they are going to go for virilization and ambiguous genitalia. So to avoid this, the definitive zone will produce excess cortisol at that period of 8 to 12 weeks. This excessive cortisol is going to suppress ACTH. This suppression of ACTH at this 8 to 12 weeks is really, really important to reduce the DHAs production during this time. So which will otherwise, if they produce excessive DHAs during the time, will result in unnecessary virilization if it's a female fetus. So this is how your fetal live adrenal gland is going to behave. As soon as the child is born, there is one important thing happens in the postnatal period. In the postnatal period, the fetal zone will involute. Plus, there will be definitive formation of this zona glomerulosa and zona fasciculata. And there will be change in the enzyme composition also and enzyme activity as well. So there will be increased activity of 3-beta-HSD, and there will be suppressed activity of 17-hydroxylase and lyase activity, which means they are going to produce more of a mineralocorticoid and glucocorticoids and less of DHEAs. Because this 3-beta-HSD is one of the key enzymes for production of this mineralocorticoids and glucocorticoids, and 17-hydroxylase and lyase enzyme is key for production of this DHEAs, which we'll discuss subsequently in the next chapter, where we'll be understanding much better when you know about the adrenal steroidogenesis. So this is what happens in the childhood, in the postnatal period, and it continues till the puberty. In the adults, let us assume the child assumes puberty. At the time of puberty, there is one thing that will happen, that again the composition, the activity of the enzymes will vary. There will be excessive ACTH production. There will be surge in the ACTH production from the pituitary at the time of puberty, which will actually increase the 17-hydroxylase and lyase activity, and there will be a little bit low activity of 3-beta-HSD in comparison with the 17-hydroxylase and lyase activity. And this is going to trigger once again more production of DHEAs and little bit lesser production of cortisol. I'm not telling cortisol production will be suppressed, but little bit lesser production of cortisol and a little bit relatively more production of DHEAs during puberty. And again, these adrenal androgens are very, very important from female perspective, not from male perspective, because in males, the storehouse of androgens and the factories of androgens comes from the testis. Because in testis, you are going to produce a lot of androgens in the form of testosterone, and which will be further converted to dihydrotestosterone in the periphery. And that itself is enough for the males, and the adrenal androgens play a very, very little role as far as a male is concerned. But for females, the principal source of adrenal androgens is, principal source of androgens is coming from the adrenal gland only. Clear? So that to this DHAs is one of the main source of androgens in the females. And in the female, at the time of puberty, they do develop this axillary and pubic hair, right? So the development of this axillary and pubic hair in a female is based on the adrenal androgens. But in males, adrenal androgens play a very little role. We don't know what role they really play because they are very weak. And in comparison to the storehouse and the factories of androgens, that is the testicles, this adrenal androgens really is nothing. And in a male, the development of male pattern is completely dependent on the testicular androgens. Clear? But in females, the development of this axillary and pubic hair and this sexual characters is totally based on the adrenal androgens, which means the adrenal androgens are the principal source of androgens in a female after puberty. Clear? Very, very important point. So this process of excessive DHEAS production at the time of puberty is what we refer to as adrenarche. In a female, this adrenarche is really, really important for this pubic and axillary hair. But in males, adrenarche is not really important for the production of pubic, axillary hair, but the testicles are important. I am substantiating this point multiple times. Let's shift our focus completely to the gonads now. So at 6 weeks, probably around 6 to 7 weeks of time, this gonad is also referred to as a bipotential gonad or an indifferent gonad, which means till 6 weeks, they have the tendency to form either a testis, or they can even form an ovary, or they can form nothing and they can vanish in the form of a gonadal dysgenesis also. So there are some things that determine whether they will become a testis in the first place or they will become ovary in the first place. So what are the things that determine the differentiation of this gonad? The first and foremost important thing is the SRY. The presence of this SRY protein coming from the Y chromosome in the SRY region of the Y chromosome is really, really, really important for development of this testis. Apart from that, there are some other downstream players like SOX9, which is also important for the development of the testis. The importance of the SOX9 will be understood when you start understanding about this disorders of sexual differentiation. Similarly, ovarian development is triggered by the absence of SRY, which is very, very important for testicular development. So you don't have SRY because in females you have X and X, and you have another regulator called a DAX1, which is very crucial, to be honest, because DAX1 is also referred to as an anti-testis factor, which means it prevents the development of the testis. And at the same time, there are other factors like FOXL2. This is also important for development of the ovary. In that, I feel like this DAX1 is very, very important as far as ovarian development and female development is concerned. So these problems can result in disorders of sexual differentiation. Like for example, if you have a patient, the genotypical male, that's a XY, it's a male karyotype. If you have a sufficient expression of SRY as well as the SOX9, you will result in a phenotypic male, which means their external genital and internal genital is going to look like a male. And you have a XX genotypical females and you don't have SRY. At the same time, you have this DAX1 where they're going to develop into a phenotypical and a genotypical female. But you have any problems in this, you're going to result in a DST. Let us assume what are the problems you might be facing. For example, you have a XY genotype, but if the patient is not having a proper SRY expression, which means due to some reason you don't have this SRY expression due to deletion or mutation, or you don't have this proper SOX9 expression. Even SOX9 expressions are also problematic. Or if you have an additional DAX1, like in this case, if you have a DAX1 duplication, because I told you this DAX1 is a very important anti-testis factor in case of females. So if you have a DAX1 duplication, extra DAX1, so all these things, or even any one of these, if it's present, it's going to not result in a testicular formation. They'll go for gonadal dysgenesis because SRY and SOX9 is important for production of testis and DAX1 antagonizes the testis. There's some problem here. Testis production will be antagonized and the testis will go for dysgenesis. In other words, we can call it as gonadal dysgenesis. And this gonadal dysgenesis will result in poor exposure of a male genotype fetus and they won't have an androgen, which means the fetus will not virilize and they won't develop the male characters. And they will look like a phenotypical female, which means these are called as XY females, or otherwise simply we can call it as XY DST. Please remember the term, you should not call the word hermaphrodite, true hermaphrodite, pseudo hermaphrodite, or you should not use the term called intersex because the current terminology is very, very clear. You have to always stick on to the term called DST, disorder of sexual differentiation. Clear? So these patients will be looking like a phenotypic females and they may come with ambiguous genitalia. And these are actually having a karyotype of XY, but the outside they look like females because they don't develop a testis and they don't have androgen exposure, so that the external genital and internal genital may not sufficiently develop. So they are called as XY DST or XY females. If you have XX karyotype and you have an additional SRY, or you have an SRY in this patient, you should not express SRY. Due to some reason, these females are expressing SRY. What is going to happen is they are going to produce testes and they are going to virilize these females, and they are called as XX males, or otherwise you can simply call it as a XX disorder of sexual differentiation, where this SRY will translate into production of this testis, which is going to produce excessive androgens, which is going to virilize the female fetus, resulting in some ambiguous genitalia, and they are called as XX males or simply XX, but as a disorder of sexual differentiation. So that is why these transcription factors are really, really important to understand. If there is any problem in the transcription factors and any problem in these factors which I have told you, will result in a disorder of sexual differentiation. So let us move on to further unique process of development, further development of the testis and development of other male structures. So what this testis is going to do, this testis is going to have two types of cells. One is the Sertoli cells and another is the Leydig cells. This Sertoli cell production is under the control of this SOX9, and this Leydig cell development is under the control of NR5A1, which I have told you already in the adrenal gland embryology itself. But apart from that, these are just testis, and you need to have the germ cells, right? So germ cells come from a completely different source. You have a region called a hindgut. So what are the germ cells? These germ cells are the ones that are going to result in future oogonia and spermatogonia, which means the sperms and the ovum are actually coming from the germ cells, not from actually the gonads, which means the hindgut is the one that is going to contain these germ cells. These germ cells, the origin is from the yolk sac, and this process also happens around 6 to 8 weeks time. So where these germ cells will migrate towards the testis as well as the ovary under the influence of a lot of transcription factors like WNT5 and SDF1, and they are going to come into the testis as well as the ovary. So I can write the germ cells inside here, and I can write the germ cells into the ovary also. But remember, this germ cell production and movement, migration, and the testicular development and further production is completely different, and they have two different branches. Suppose you can have a normal testis or ovarian development, but complete absence of sperms or complete absence of oocytes inside the testis and the ovaries respectively, because these germ cells may not migrate properly. So a patient who's having a normal testis, normal external phenotype of a male, and an XY karyotype can be completely infertile if the germ cell migration is poor. So that is why this germ cell migration is really important to understand. It's a completely different process which governs this movement of the germ cells into the testis, which are the future reasons for producing a spermatozoa and oocytes. Clear? And there are some germ cells which escape this migration. Usually these germ cells that escape this migration are generally going for apoptosis. They are going to go for apoptosis. But in rare instances, some germ cells may escape this migration process and apoptosis process, and they may not enter the germ cells, and they also escape apoptosis, and they can deposit in the mediastinum, brain, or even in the abdomen, which is going to be the source of future germ cell tumors. They are going to produce, this HCG, these are called as HCG producing germ cell tumors in the adult life. Clear? That is due to this escaped germ cells that have escaped this apoptosis in the embryonic life. Clear? So as I told you, the testis producing this Sertoli, differentiating into Sertoli cells and Leydig cells, depends on these two important transcription factors, that is SOX9 and NR5A1. The Sertoli cells, they are going to produce two important factors. One is called inhibin B and second is anti-Mullerian hormone. Other is called as Mullerian inhibitory substance. and these Leydig cells are going to produce testosterone, and they are also going to produce this insulin-like factor 3. That's called INSL3 or insulin-like factor 3. These are the two important things that are secreted by these Leydig cells. Similarly, the ovarian production and sustenance is also very important. It's mediated by a number of transcription factors, like you have WNT4, you have FOXL2, and you have DAX1. All these things we have studied already, and all these things are going to sustain and maintain the ovarian development. And obviously in the future, these germ cells are going to become spermatozoa as far as testis is concerned. And as far as your ovary is concerned, these germ cells are going to become oocytes. How the same germ cell is becoming two different type of cells, is differentiating into two different type of cells, different in a male and different in a female. Because this X chromosome is going to produce another factor that's called a STRA8 or STRA8, which is something like a retinoic, it acts on the retinoic acid receptor. And this is the one that is going to differentiate this germ cell towards the formation of this primordial follicle during the embryonic time. I'll be discussing in the next section on the reproductive physiology, where I'll be discussing more on the primordial follicle development, which happens in the females during the embryonic life itself. This is nothing but a oocyte. And a primordial follicle development means, primordial follicle means they will have oocyte surrounded by a flat granulosa cells. Clear? So, and STRA8 is very, very important for differentiation into a oocyte and a formation of a primordial follicle in the ovary. And males do not have this STRA8, which means the germ cells will not develop into a primordial follicle and a oocyte. Instead, in the future, they will be forming a spermatozoa and spermatogonia. So, the Sertoli and Leydig cells, what are they going to do? So, for that, you have to understand what are the different ducts and different organs that you see in the embryonic life. So, you have something called a Mullerian duct or a paramesonephric duct, which is going to result in a female internal genitalia structures. Like for example, you will be forming uterus, cervix, and probably upper part of the vagina and the fallopian tubes. These are generally formed by the Mullerian ducts. And you have Wolffian structures. These are called as mesonephric ducts, which forms the male internal genitalia structures. At the same time, these Wolffian ducts also gives rise to, otherwise called as a mesonephric duct, this also gives rise to the ureters and the renal pelvis in both sexes. But we're not going to discuss about that. The Wolffian duct in males is important for the formation of male internal genitalia, like your seminal vesicles, vas deferens, and epididymis. Clear? At the same time, you have to know the development of the external genitalia also, which I'll be discussing subsequently. So, but what really happens in a male is, because the Sertoli cells generate this anti-Mullerian hormone, they are going to be anti-Mullerian and they are going to block the development of this Mullerian duct. That is the reason why you don't develop this Mullerian duct structures in a male patient. Which means this process is what we refer to as an active regression, because this Mullerian duct, otherwise if you don't have this AMH, they will grow spontaneously and it's a passive thing. And similarly, this Wolffian duct, to sustain, you need this testosterone. This testosterone is needed to sustain this Wolffian duct. So this testosterone in a male that is coming from a Leydig cell from the testis is going to continuously stimulate this Wolffian duct to sustain and develop this male internal genitalia structure, so that a male will develop this male internal genitalia structures. Whereas in a female, something else will happen. Like for example, females also will have Mullerian ducts, which is going to result in formation of female internal genitalia, and they have Wolffian ducts as well, which are going to result in a male internal genitalia. In a female, you know this ovary does not produce testosterone, which means if you do not have this testosterone, this Wolffian ducts need continuous stimulation of the testosterone to sustain. So if you do not have this testosterone, you cannot progress with this Wolffian ducts, and the male internal genitalia structures will not be produced in a female. Similarly, if you do not have testosterone, this Mullerian ducts passively develops into a female internal genitalia, and this regression of Wolffian ducts because of absence of testosterone is what we refer to as a passive regression. Remember, testis actively secretes a hormone to regress the Mullerian ducts. That's called active regression of Mullerian ducts happens in the males. But in the female embryo, there is a passive regression of Wolffian duct because even without testosterone, the Wolffian ducts will passively disappear because of the absence of testosterone, just because of the absence of testosterone. And no testosterone, automatically the Mullerian ducts will develop into female internal genitalia by default, which means they are going to develop into uterus, your fallopian tubes, cervix, and the upper part of vagina. Remember, the lower part of vagina is a external genitalia, which comes from the urogenital sinus. It is also worth noting the fact that to produce a female type external genitalia, you don't need anything, which means you just need absence of testosterone. This is what happens in any embryo and any fetus. If you do not have testosterone, and for that matters, this is going to convert to dihydrotestosterone, which means you don't have any exposure to androgens, or in general, I can write absence of androgens. This is enough to produce a female type external genitalia. In fact, even female type internal genitalia as well, which means females just need no interference from males. That's what they need. If they don't have any interference, they are going to transform into a female type external genitalia and internal genitalia. So what do you mean by a female type external genitalia? You have no labioscrotal fusions. I'll be discussing about this labioscrotal swelling in some time. This absence of labioscrotal fusion is what is going to, get into an open type configuration in a female, where you'll have a labia majora, and this urethral folds will not close. That will result in a labia minora. At the same time, they'll be having only a clitoris, but they won't have a penis. Then they'll be having a single urethral opening. Clear? So this is what is happening in a female type external genitalia, because they just need no androgen influence. That's all. They will develop into a female type internal genitalia and female type external genitalia. But in males, this external genitalia formation is something that is completely dependent on your testosterone. So this testosterone is going to be converted to dihydrotestosterone in the target tissues by an enzyme called 5-alpha reductase, otherwise referred to as 5-alpha reductase 2, intracellularly in the target organs, where this dihydrotestosterone tends to be almost 3 to 4 times more potent than the testosterone itself. What is the importance of this dihydrotestosterone? Because this testosterone as well as dihydrotestosterone both can act on the androgen receptor. But one crucial point to note is the fact that this testosterone alone is enough to develop male type internal genitalia, which means to develop that seminal vesicles, vas deferens, and epididymis, you just need testosterone, nothing else. But testosterone is not enough to develop this external genitalia for male type, because testosterone, for converting this internal genitalia to male type, they just need to act in a very shortcut. Like this internal genitalia is central organs, and testosterone also released from central area, so they are very close by. So the action is easy. But for external genitalia stimulation, they have to go systemically into the systemic circulation. They go outside and they have to stimulate the cutaneous receptor. By the time they go systemically, there could be a lot of metabolism of testosterone resulting in very poor concentration. And since being weaker compared to DHT, this poor concentration of testosterone is not enough to convert this external genitalia into male type external genitalia. To produce this male type external genitalia, you need this dihydrotestosterone. Even at lower concentrations in the systemic circulation, they are very potent, so they can stimulate the cutaneous receptors and they can convert this external genitalia into male type external genitalia. So which means to produce a male type external genitalia, which is classically defined as labioscrotal fusion resulting in formation of scrotum, and there will be fusion of urethral folds, that is going to result in the median raphe, and obviously they are going to result in a single urethral opening because of the fusion of urethral folds. So, but this labioscrotal fusion is a classic hallmark of a androgenic influence. Clear? Any androgen exposure, especially dihydrotestosterone or any other androgen in excess, can result in labioscrotal fusion. Even in a female, if you expose this female children to this androgens, there will be labioscrotal fusion resulting in ambiguous genitalia in a female child. But in a male child, this process is completely normal, and this is what we refer to as a virilization, which means virilization is a normal process as far as a male child is concerned. But in female child, they should not be virilized, because virilization is the term that we use for exposure to androgens. Females should not be exposed to androgens, and there will be no virilization happening in a female child or in a female embryo. Males, virilization is a normal phenomenon. So they have to be fully virilized at the time of birth because of this androgenic influence. Clear? So that is the basis of this XX and XY DSDs. Usually this XX DSD means you have basically a genotypically female, because genotype is XX and these are genotypically females, but they are presenting with the ambiguous genitalia, which means they are virilized. Normally a female child should not be virilized, but because of the virilization, they are presenting with ambiguous genitalia. Obviously, I told you this virilization means it's a sign of an androgen exposure. So what do you mean by that? Which means whenever you have a XX DSD in a female child, it's always due to excess androgens or androgen exposure, and this is what we refer to as an act of commission. Which means whenever the females commit towards androgen exposure, they might get virilized and they might be born with ambiguous genitalia, and this is the basis of the XX DSDs that you're going to get. What is the base of XY DSDs? This XY DSDs, obviously they will be genotypically males, which means they will have XY karyotype, but they will be under-virilized. Because of that, they will be having ambiguous genitalia, which means their labioscrotum should completely fuse with the exposure of adequate amount of androgens. If they do not have this adequate amount of androgens, their labioscrotum will not fuse properly, and they also might look like an open configuration at a female type, and that's what we call it as a ambiguous genitalia, and this is because of under-virilization of a male fetus. This under-virilization is due to either poor androgen exposure or due to androgen resistance, like in complete androgen insufficiency syndrome, where they might look like a completely phenotypical females. So this is an act of omission, which means the males omit from exposure to androgens, where they may result in under-virilization and formation of ambiguous genitalia. So DSDs, XX DSD is due to act of commission, where there will be androgen exposure, and XY DSD is due to under-virilization, and it's an act of omission because of poor androgen exposure. These are the basics of disorders of sexual differentiation, which we'll be discussing in the subsequent videos. So I also need to substantiate one more important clinical point about this 5-alpha reductase 2 deficiency, because you will be encountering a common terminology called 5-alpha reductase deficiency. What do you mean by this? The basics I'm going to discuss in detail later on. So whenever you don't have a 5-alpha reductase deficiency, you're going to produce very low levels of dihydrotestosterone, but there will be a good amount of testosterone. But I told you in the embryonic and fetal life, your testosterone alone is enough to produce a male type internal genitalia, but they are not enough to produce male type external genitalia, which means these patients with 5-alpha reductase deficiency, they will normally produce this seminal vesicles, vas deferens and epididymis, but they cannot produce this male type external genitalia, which means they will have under-virilization because of poor DHT exposure, and this will result in probably, if it's depending on a complete or a partial 5-alpha reductase deficiency. If it's a complete 5-alpha reductase deficiency, the external genitalia will never be virilized and they will look like completely a female type external genitalia, and they will be reared like females. If it's a partial 5-alpha reductase deficiency, they'll not look like a female or they'll not look like a male, because they'll not be complete virilization, there'll be only partial virilization, and they may come present, they may be born with the ambiguous genitalia. Clear? But a complete 5-alpha reductase deficiency will usually result in a female type external genitalia at birth. So usually these children will be reared as females till puberty, but there is something dramatic that happens during puberty. So what happens during puberty is the fact that there will be pubarchy. Pubarchy means excessive production of FSH and LH from the pituitary gland. This normally happens, and this excessive FSH and LH is important for production of this excessive testosterone. They'll produce too much amounts of testosterone. Now, this much high level of testosterone might be sufficient in these patients to virilize this female external genitalia at the time of puberty to become a male type external genitalia, which means this history of virilization during puberty is one of the classic pointers towards 5-alpha reductase deficiency in exam. Remember the fact I already told you that maternal virilization during pregnancy is a very classic pointer towards aromatase deficiency. Similarly, your virilization during puberty of a female looking child might be a very important pointer for 5-alpha reductase deficiency, especially in exams. Do not forget this point. And the next thing what you have to know about the male children, which is very unique to the male fetus and embryo, is the fact that they go for something called testicular descent. And this testicular descent is based on these two very important things, that is testosterone and insulin-like factor 3, which is coming from the Leydig cells, which in turn is coming from the testis itself, because the testis itself is responsible for its own descent into the scrotal region. So I can write this testosterone is important, at the same time this insulin-like factor 3 is also important for descent of the testis. But up to what level? This insulin-like factor 3 is the main reason for the movement of the testis from the abdomen up to the inguinal region, and this testosterone is important for the movement of the testis from the inguinal region to the definitive location, that is the scrotum. So what is the clinical aspect of this? So in a condition where you have an entire testis problematic, like you have a dysgenic gonad, we call it as a gonadal dysgenesis. Gonadal dysgenesis, where you do not have this insulin-like factor as well as testosterone. So there won't be any descent of the dysgenic gonad, and they will look completely bamboozled, and they will have an abdominal testis, which means they will not have any sort of descent. So whereas in conditions where you have problems with testosterone, like testosterone deficiency, like 17-beta-HSD3 deficiency, which we'll be discussing later on in the subsequent videos, or if you have a testosterone resistance, androgen resistance, like androgen insufficiency syndrome, in these conditions you will have an inguinal testis, because in this condition the testis is present, but there is some problem with the testosterone, not the testis, which means the testis will normally produce this insulin-like factor 3, which can pull the testis up to the inguinal region, but they cannot go to the scrotum because of the testosterone deficiency or resistance. So you'll have an inguinal testis here. And very, very important point is the fact that this abdominal testis has a very, very high risk of malignancy compared to that of the inguinal testis, which has a very, very low risk of malignancy. Very, very important point. Clear? So remember, final take home point: dysgenic gonad, abdominal testis, testosterone related issues, or androgen resistance, it will result in inguinal testis. Clear? And final importance of this external genitalia lies in the fact that there is a window period called 8 to 13 week window. This is a final thing I want to tell, and this is a very crucial thing to understand. There's a 8 to 13 week window period, which is really important for the external genitalia to form, because any abnormality after this 13 weeks, any abnormality that resulted in external genitalia cannot be reversed. That's the most important point. You can underline the fact that it cannot be reversed. Very, very important after 13 weeks. During this window, it's a very crucial time, which means a male child without androgen exposure will have a small phallic size. And once you have a small phallus in a male child, after 13 weeks, it cannot be reversed. Very important. At 13 weeks in ultrasound, if you have a small phallus and it's a male child, in amniotic fluid sampling or chorionic villus sampling, it is definitely not reversible. Similarly, if a female child by genotype with androgen exposure, if it has virilized already, which means there is a labioscrotal fusion. This is a classic example of virilization, labioscrotal fusion. After 13 weeks, again it cannot be reversed. This is the basis of giving prophylaxis dexamethasone in congenital adrenal hyperplasia, but before 8 weeks. So I'll discuss with you in the CAH section. Don't worry about that. So that's why I'm telling you, after 13 weeks, any abnormality, if it happens, it cannot be reversed in the first place. That's a very, very crucial point. And the second point you have to understand is the fact that this 8 to 13 week window, your androgen production, androgen production is very important for males, isn't it? Females, nothing is needed. They grow by default. It's passive. That's what, is the traditional understanding, and even though many new theories are coming, but traditional understanding, let us go by that. The traditional understanding is the fact that females don't need anything. They grow by default. But they just should not have any androgen influence. But in the males, you need this androgen influence. That's why I always called males as corrupted females. These are nothing but females corrupted with Y chromosome and androgens becoming a male. Fine. So androgen production. So once you have produce this androgens, you are going to produce this virilization. But it's very important to understand the fact that this androgen production in males in this period of 8 to 13 weeks is not dependent on pituitary. Not dependent on FSH and LH. That means it's not dependent on pituitary, FSH and LH. So then how can you produce this testosterone in a male child? Because we know that Leydig cells are generally, in physiology you would have studied, they are stimulated by your LH only, luteinizing hormone. Then Sertoli cells are stimulated by FSH. We know that. But how in the world without FSH and LH you can produce androgens in this 8 to 13 weeks period? It's very, very important point, because at this 8 to 13 weeks period, your androgen production in a male child is dependent on beta HCG from the testis, from the Leydig cell, not based on the FSH and LH. That's why the beta HCG rise in the late first trimester. It peaks in the late first trimester at around 10 to 12 weeks mark. This has multiple implications, not only on the thyroid perspective, on the growth of the external genitalia in a male child perspective also. Clear? But remember, females, this beta HCG will not have any effect, because females produce ovary by that time. And this beta HCG has no influence on, because females don't have Leydig cells, and this beta HCG is not going to stimulate anything in the female child. So let us see what's happening and how this mechanism works. You have a Leydig cell, and the Leydig cell you have a receptor called LHCG receptor. That's called common receptor for luteinizing hormone as well as the human chorionic gonadotropin. Because this beta HCG has alpha subunit that is similar to TSH, FSH as well as LH. So which means this beta HCG can work like an LH on the LHCG receptor in the Leydig cell. I can write this as beta HCG and act on the LHCG receptor, and they are going to stimulate the testosterone production. Clear? Why this point is important? Means even if you do not have this entire pituitary, for example, in patients who are having congenital hypopituitarism, like for example a Kallmann syndrome, they don't produce any gonadotropins, FSH and LH throughout their life. It's a congenital hypogonadism and hypopituitarism. But remember the fact that in patients with Kallmann syndrome, they have a normal looking external genitalia, which means there will be normal labioscrotal fusion, there will be normal penis, and there will be normal looking external genitalia. But the only problem in this patient will be FSH and LH are very important for production of testosterone after 13 weeks, but production of testosterone after 13 weeks is important only for enlargement of the penis. That's it. Which means this patient will have micro penis only, nothing else. They just have a micro penis. Apart from that, the external genital looks typically like a male only. That's a very important point. Even in a pituitary problem, pituitary agenesis, complete absence of pituitary, or if you have a congenital pituitary issues like Kallmann syndrome where you don't have any gonadotropins, still you develop a normal looking external genitalia because of the beta HCG, not because of the FSH and LH, and only problem these patients will be having is micropenis. Similarly, in a female child, I told you after 13 weeks, you cannot reverse. At the same time, it has a positive implication also. After 13 weeks, if you have androgen exposure, it's a XX, but after 13 weeks, you have androgen exposure. After 13 weeks, they won't be virilized. They'll be looking like a female looking external genitalia only. There will be no virilization, there will be no labioscrotal fusion, because that 8 to 13 week window is the window, like beyond that you cannot correct anything. You cannot do anything even if you expose them to androgens, which means there will be no labioscrotal fusion. Just they will have only one thing, that is clitoromegaly. Even if they are exposed to androgens, they will have just a clitoromegaly, which is in males, the clitoris counterpart is the penis in males. So here, like how you have a little bit of a small penis because of poor androgens in males, because of testosterone deficiency after 13 weeks. Similarly, after 13 weeks, if you have a androgen exposure in females, you will just result in a clitoromegaly, nothing else. Otherwise, there will be no labioscrotal fusion and they will be looking like a normal appearing female external genitalia. Clear? So this is the very important understanding behind this 8 to 13 week window period, where everything happens with regards to external genitalia, and it's so crucial to understand this 8 to 13 weeks window period. This is also one of the basic reasons why we tend to correct this congenital adrenal hyperplasia, especially 20 alpha hydroxylase, especially it's a female child. We tend to correct this or prevent this virilization before this 8 to 13 week window period itself, where they start with steroids before this 8 weeks, around 6 weeks marks itself, if both the parents are carriers for 20 alpha hydroxylase deficiency. So we'll discuss that later on. So that this forms the basis of that, because after 13 weeks, you cannot correct. At the same time, after 13 weeks, even if you expose them to androgens, the female child will not get virilized. Clear? So this is the basics, and let us go and see some of the images. This is only for the completion sake. So I told you by around 6 weeks time, you will have a bipotential gonad, where the gonad might go towards testis or as well as a ovary. That time you have a mesonephric duct, which is otherwise called a Wolffian duct, and you have a Mullerian structure called as a Mullerian duct. Both will be there. In a male who's going to have a XY, your presence of SRY will result in the formation of testis, and testis, along with the production of testosterone and dihydrotestosterone, they are going to result in male internal genitalia and external genitalia. So I can write testosterone here, which is going to result in male internal genitalia, and the testis is also going to produce anti-Mullerian hormone that will result in degeneration of the Mullerian structures and growth of the Wolffian structures into vas deferens, seminal vesicles, and epididymis, and they are going to result in this testosterone is going to sustain this Wolffian duct structures. This anti-Mullerian hormone is going to cause degeneration of the Mullerian structures. Females, nothing much. You have just XX, which means there is no Y, which means no SRY. And other transcription factors, as I told you, formation of ovaries. Once your ovaries are formed, you don't have anything, there is no testosterone, that's enough. And there is no dihydrotestosterone, that's enough. And there is no anti-Mullerian hormone, that's more than enough. And because there is no anti-Mullerian hormone, there is nothing to regress the Mullerian structures. So obviously the Mullerian duct will grow into uterus, fallopian tubes, cervix, and upper part of the vagina. And because there is no testosterone, the Wolffian structures will degenerate and they will disintegrate. You can see there will be degeneration of the Wolffian duct here. The Wolffian duct will degenerate and disappear here. Because there is no testosterone, you cannot sustain the Wolffian duct. So this is the basics that are happening in the embryological life. Then what about the external genitalia? External genitalia is composed of multiple structures, like you have, this is the indifferentiated external genitalia. This is sexually indifferent stage by around 5 weeks. Remember, the first time where you can actually identify the sex of a child through ultrasound is around 10 weeks. That's why it's best after 13 weeks, because that 8 to 13 weeks period is the cutoff where you develop this external genitalia concretely. So after 13 weeks in ultrasound, concretely you can diagnose the sex of a child if you want to take in India, even though it's banned. But minimum it takes 10 weeks. Before 10 weeks, it's very difficult to concretely tell what is the sex of the child. So especially at around 5 weeks, you can see it will be a completely indifferent external genitalia. These are common things where you see a labioscrotal swelling and genital tubercle and a urethral fold, anus and urethral groove. Where in a male, because of the actions of dihydrotestosterone, nothing else, everything will happen. Like you're going to result in the formation of glans penis, where you from where you form from the genital tubercle. This is going to form the penis and the glans penis, and this labioscrotal fusion is going to form the scrotum, and this urethral folds is going to close, resulting in a single urethral opening. And finally, you will result in a male type external genitalia. Everything is due to dihydrotestosterone, glans penis, scrotum, anus, and this fusion of the labioscrotal region and urethral folds is going to come like a median raphe. You can see in male adults and children. In females, the idea is very simple. You don't have dihydrotestosterone. That's it. Or you don't have any androgens for that matters. No androgens for that matters. So which means nothing is going to happen here. Their genital tubercle is going to remain the same as clitoris, that's glans clitoris, and your labioscrotal swellings is going to be open just like that, resulting in a labia majora. And the urethral folds are not going to close as well, resulting in the formation of labia minora. And finally, this is a adult female or a female child where you're going to have a glans clitoris with the labia majora and a labia minora. Nothing will close. It will look just like the same. And these are this is a very important table that you have to know as far as which structure results in what structure in male as well as the female. You can see the gonads, depending on XX or XY, and depending on multiple factors, they're going to become ovary and the testis. The cortex is going to become, of the gonad is going to become the seminiferous tubules. Here the ovarian cortex will become follicles, and the middle of the testis will become retitestis, and here it will form the retiovary or the stroma, and the gubernaculum will become gubernaculum testis, and in females it will become ovarian ligament and the round ligament, and mesonephric tubules in males will become efferent ductules, in females will become epiphoron and paraphoron, and the mesonephric duct, which is very important for exams, which is going to form the male structures, right? So the epididymis, vas deferens, seminal vesicles, these are very important male structures, whereas females, there is no much use. At the same time, the mesonephric duct is important for both, for the formation of ureter, pelvis, and the collecting duct. So this is common for both, which means from the internal genitalia perspective, this mesonephric duct is not important for females. But from kidney perspective, like this mesonephric duct is still giving rise to this ureter, renal pelvis, and the collecting duct and the collecting system in both males and the females. And you have a paramesonephric duct. This is otherwise referred to as a Mullerian duct, which is very, very important for female internal genital structures like uterus, fallopian tubes, this upper part of the vagina, and the cervix. And in males, it's not that important. They're going to form a vestigial structure called appendix of the testis. That's not so important. And this urogenital sinus is a very important external genitalia structure, whereas in both males and females, it will result in bladder and urethra for sure. But remember, in males, these are the ones that are going to form the prostate gland. That's very important. The urogenital sinus is the one that forms the prostate gland. In females, the urogenital sinus is the one that forms the lower part of the vagina. That's really important because upper part of the vagina is formed by the Mullerian duct. Apart from that, they form this urethral and paraurethral glands. For example, Skinner's glands, which are analogous to prostate in female, but their function is unknown. But the most important point in female is the urogenital sinus forms the lower part of the vagina. That's the crucial point as far as exams is concerned. And genital tubercle, we know in male they form the penis and the glans penis, whereas in females it forms the clitoris and the glans clitoris. And urethral folds, they form the ventral aspect of the penis in case of males, whereas they form the labia minora and they won't be closing. And the genital swellings, they form the scrotum. And genital swellings, in this sense, we are calling this as labioscrotal swellings. These labioscrotal swellings in males, we know they're going to form the scrotum by labioscrotal fusion, and in females that will be open as labia majora. So these are the major structures with regards to the embryology that you have to know regarding the female and the male external genitalia. I think with this, we have completed the demonstration of embryology and its importance in understanding about the DSDs, because DSDs is a huge topic. It needs understanding of steroidogenesis, which is going to be discussed in the next section, and a good understanding of reproductive physiology like menstrual cycle is also important, which will be discussed in the subsequent chapters. And after that, we'll be discussing on the congenital hyperplasia. Then we'll go back to the DSD so that you'll be understanding much better. So this is how I plan, and let me summarize whatever we have told. So first important summary is the understanding of DSDs, how you are going to develop a disorder of sexual differentiation. It could be a XX DSD or a XY DSD. We know XX DSD is due to act of commission, where you have the female fetus committing to excess androgens. Any level of excessive androgens will result in ambiguous genitalia and a disorder of sexual differentiation as far as the female child are concern. And in a XY DSD, it's due to usually act of omission, where it is due to poor exposure or non-exposure to androgens that will result in under-virilization of a male fetus, resulting in an ambiguous genital and a XY disorder of sexual differentiation. And you also know that this 8 to 13 week window is very crucial, and any disorder after this 8 to 13 weeks window period cannot be corrected, or otherwise if the external genital is formed normally at this 8 to 13 weeks, after this 8 to 13 weeks mark, either you have over androgens in a female child or less androgens in a male child is not going to affect the male child in a much bigger way. So this is very important. At the same time, you have to note that point regarding this testicular descent, where there are two factors that are important. Remember, in a completely dysgenic gonad in a male, you will have an abdominal testis where you have a testosterone deficiency or resistance to androgens. You will result in inguinal testis rather than abdominal testis. And more importantly, I've also mentioned about this testosterone influence on gender identity in the beginning, but I have not explained in detail. But this testosterone is very, very important for gender identity as well, which means this testosterone influence on the brain at the fetal life. There are a lot of enough evidences for that. The fetus is influenced by testosterone at 8 to 13 weeks mark itself, in the first trimester itself, where the fetus starts to think like a male. So which means there are certain male activities, even though this is not related to chauvinism, this is not related to anything, this is purely related to hormones. A male thinks like a male. So they have conducted a lot of experiments where when they infuse testosterone into a female, like rat, where they started behaving boisterously, like very hyperactive, they started bossing over the other rats. So this is a male type behavior. So this influence of a male type behavior starts at the embryonic level itself due to exposure to testosterone. That is the reason. This explains why there is a poor exposure to androgens, even though you don't have any DSD, but in the later life, these male children, because of some reason of poor exposure of testosterone to the brain, they start thinking that they are females, gender identity disorder. Similarly, a female child may be born as a female child, because of some reason they're exposed to androgens in the brain at 8 to 13 weeks mark, they start thinking that they are males. And this testosterone has a huge influence on converting this brain to think like a male. And that's clearly proven by many experiments, and testosterone is very, very important for this gender identity as well. And finally, two important points: whenever you have a history of maternal virilization during pregnancy, it is a straight pointer towards aromatase deficiency, even though there could be a P450 oxidoreductase deficiency. I don't want to confuse you, because P450 oxidoreductase is a separate class of enzyme that is important for three enzymes in the adrenal steroidogenesis: 17-hydroxylase, 21-hydroxylase, as well as aromatase. So three enzymes that are united together by that P450 oxidoreductase. So you can get this maternal virilization history in a P450 oxidoreductase deficiency also. But in exam, please do remember this aromatase deficiency. Whenever you have a history of virilization in a female looking child at puberty, always think about 5-alpha reductase deficiency in exam. This is a very, very direct pointer in exam, where the examiner wants to test you about this. Whenever you have a history of virilization at puberty, think about a 5-alpha. There are a lot of causes are there, but you think about 5-alpha reductase 2 deficiency in exam. With this, my summary is over. These are the net crucial points that you have to note for exams, and I've explained to you in detail about how this embryology happens, and all these are very crucial in your future understanding of disorder of sexual differentiation. With this, we'll sign off with the current video, and we'll move on to the next video, which will be on the reproductive physiology.